Okay, good afternoon. Everybody's very welcome. We've got a special seminar today. In EEB, we have a tradition of newly promoted and tenured colleagues are invited to give a talk in celebration of their promotion. And recently, our colleague Dan Rabosky was promoted to Associate Professor in EEB and Associate Curator of Herpetology. Well done, Dan. Let me give you a very quick bio of Dan, and then we'll let him talk. So Dan got his Bachelor of Science at Ohio University, his Master's in Pennsylvania State University, his PhD at Cornell. He followed that up with a 3-year Miller Research Fellowship in Berkeley, and then we successfully recruited him to join us in 2012. So as you can tell from his title, he's really big into biological diversity, and Dan doesn't do things by any half measures. He's into it in an epic way, a big way. Not just the scale of the work he does, but he takes a multidimensional approach. Many biologists and scientists generally split into either theoretical, people who are interested in theory, or empiricists. He blends the two in very creative ways. He's probably best known for his theoretical contributions for developing a very influential computer program called BAM, or Bayesian Analysis of Macroevolutionary Mixtures. This is used to detect variation in rates of evolution across phylogenetic trees. It can look at things like rates of speciation, rates of phenotypic evolution, amongst many other features. And in his empirical contributions, they are legion. They are typically in very high-profile journals. They often involve extensive collaborations, and they are very large in scale. Just to give you one recent example, last summer he led a Nature paper on titled "An Inverse Latitudinal Gradient in Speciation Rate for Marine Fishes." That's just one of many, many papers he's published. Dan has won almost all major awards you can imagine for somebody at his career stage. Very briefly, since he joined us, he's been awarded the Dobzhansky Prize for the Society of Study of Evolution, the MacArthur Wilson Award from the International Biogeographical Society, a highly prestigious Packard Fellowship for Science and Engineering, and on our campus here he's won a highly prestigious Henry Russell Award. So the title of his talk is Species, Speciation, the Origins of Biological Diversity. Can you please join me in welcoming him, and we're ready to hear from you.
Can everyone hear me okay? Great. Thanks a lot for that intro. So it's really nice to be back here giving a talk. Well, actually, given that I gave my job talk here in fall of 2010, it's been quite a long time since I gave a formal departmental seminar here. But today what I'm going to talk about is species, speciation, and the origins of biological diversity. It kind of ties some threads together that we've been working on in my lab. It relates to some of my long-term interests in general patterns of biodiversity. So before I start, I want to ask you to think for a minute about this question of what is speciation. So what does speciation mean to you? So if you think about what it means to the concept of speciation, then what does it mean to study speciation, and how would we go about studying speciation? And I would guess that for at least half of this room, something about reproductive isolation between lineages pops into mind. So this is basically going back to the biological species concept. And if we want to think about a textbook definition of speciation and how we study speciation, we can go to this typical widely used textbook for evolutionary biology. This is Futima. And Futima defines the central question in speciation as the central question in speciation must be how genetically based barriers to gene exchange arise. And I'm actually getting some feedback in the back there. Am I too loud? Turn this down just a little bit. All right, hold on a sec. Just have a volume on it.
On the actual unit. I turned it down here as much as I can.
Yes.
So
right.
you're just projecting a lot.
That's not going down, is it?
Well, it did go down all the way.
Is that? Seems locked, doesn't it?
That's as good as we can do. So.
Well, maybe I'll move it further away from mine.
Just that since it was going up.
Is this any better? You can still hear in the back? If you can't hear in the back, let me know. All right. So, central question in speciation is how genetically based barriers to gene exchange arise. Now, I'm going to pick apart for the next few slides a little bit of an example, and I'm going to pick apart, maybe might even say pick on, an icon of speciation research. So this is a three-spine stickleback, Gasterosteus aculeatus. So this is a sort of a famous model system in speciation studies now, and it's so famous, in fact, that you could look at a recent book on speciation, like Patrick Nozzle's book on ecological speciation, and you find a three-spine stickleback doing something over a nest on the cover of this book. So this is a famous study system for speciation research. The reason it's famous and the reason you're probably familiar with it is because when you put sticklebacks and you drop them into little ponds on the western coast of North America, you tend to get adaptive divergence into two ecomorphs, a benthic form that lives on the bottom that has one type of morphology, and a limnetic form that lives in the water column and has a different morphology. The details here aren't that important. Why it's famous for speciation is because that ecomorphological difference is correlated with reproductive isolation. So this natural selection basically drives divergence between into these two morph forms, which are then reproductively isolated from each other. And you can even take morphs that have evolved in the same direction from different lakes and put them together, and they will actually successfully spawn. But when you take different ecomorphs that evolved from the same ancestor in the same lake, they have a low probability of spawning. So ecophenotypic divergence translates into reproductive isolation in this group, and this has basically led to a giant industry in terms of cranking out many, many papers and many insights into reproductive isolation using this particular system. Now, sticklebacks really are a great speciator, and what you might not know is that for these famous sticklebacks that have been popularized from Vancouver Island, Dolph Schluter's work, and so on, there are many, many more populations around the Pacific Rim that have done the same thing. So if you go to Queen Charlotte Island, or what's also known as the Haida Gwaii archipelago, and you look at you'll find stickleback populations in this archipelago where within each watershed within this island system, you'll basically find distinct phenotypically divergent populations of sticklebacks. And so there's more than 50, estimated up to 100 populations of distinct sticklebacks in that archipelago, many of which could be recognized as biological species. And all these populations are less than 12,000 years old since the last glacial maximum. And so some folks have even taken to considering this an actual adaptive radiation. So here's a paper that was published just last year called Looking at Genomic Divergence in this Adaptive Radiation of sticklebacks within this island system. Now, despite what I just told you about there being dozens of these populations of phenotypically divergent, partially or fully reproductively isolated forms in these lake or river systems throughout the Pacific Rim, there's only 16 taxonomically distinct recognized species of stickleback. And so in the entire family, the Gasterosteidae. And so if you were to look at a phylogeny of the sticklebacks using taxonomically recognized species, you would see something that looks like this. And note the time scale here. So this is just a partial phylogeny of about 6 species. But the basic idea here is that it's about a 5 to 12 million year waiting time for each new species of good recognized species of stickleback in this phylogenetic framework. However, if you were thinking about all of the divergences among these populations that are around the Pacific Rim and elsewhere, you could actually include potentially hundreds of recently diverged populations that would qualify as species under the biological species concept. And this argument has been made in the literature. And in fact, if you were to jam these species into this phylogeny, you would get something that looks like this. This is basically what your phylogeny would look like. And I want you to pay attention to the shape of this, because this is a very interesting looking phylogeny. Because what you can see here is that you have these deep divergences here going back to in the tree. But then if you look right towards the present, you can see this explosion of diversity where the divergences are so shallow that you can't even see branch lengths, because all these populations are less than 20,000 years old in terms of their origins. If you were to actually take this phylogeny and estimate rates of species formation from this phylogeny, the speciation rates are so fast that almost every animal species would be a stickleback. And in fact, without extinction actually eliminating species, you would basically have more species in about 50 million years. You would have more species of stickleback than there are particles in the known universe. That's how fast those implied rates of speciation would be. Now, what I'm going to try to convince you of is that our exclusive focus on reproductive isolation and speciation research basically overemphasizes shallow population divergence like this. And yet almost none of these new species are actually going to persist through deeper timescales. And I'm going to argue And I think some folks will agree with me that when we think about the origins of biodiversity, the significant kinds of biodiversity, the biodiversity that we see in our backyard, the trees and plants and birds that are sitting around us right now, we're actually talking about those kinds of speciation events here. So we're interested in these deeper splits that are the speciation events that contribute the most to biological diversity. So when we think about this question of what is speciation, you might think, well, okay, speciation, reproductive isolation, we're looking at this. But I say that speciation has to be a more general phenomenon in terms of how we study it, and which we're also interested in is not just how you get this, but how this translates into this ultimately. And this is what I'm going to talk about today. And so first I'm going to start out by describing a more general view of speciation that actually goes back to Ernst Mayr, which is a little ironic because Ernst Mayr is the originator of the biological species concept, but he was actually very thoughtful about other processes other than reproductive isolation per se that could affect the speciation process. And so in Mayer's 1963 book, Animal Species and Evolution, he actually lays out the logic of a series of population-based controls on the speciation process. And one of the first things that, it's obvious that when we have speciation, populations have to become isolated through some means, physically isolated, unless you have sympatric speciation. But then you can imagine that you have the evolution of genetic differences in those populations that are genetic-based barriers to gene exchange. So this is reproductive isolation. Now, a third aspect of the speciation process in Mayer's characterization is the idea that those populations have to persist. So populations split, and they have to persist for long enough to actually evolve genetic barriers to gene exchange. And finally, Mayr argued and felt very strongly that we needed to see ecological divergence between populations as well, not because that ecological divergence was important for reproductive isolation, but because he felt that reproductive isolation was critical for the persistence of new species. And so he felt that these ingredients together translated into what he called successful speciation. We've reviewed some of this work with a couple of my recent postdocs in a paper we have in review now. Now, he's got a whole chapter in this book on the ecology of speciation, which is quite different from a view that you would get from mainstream ecological speciation research today. It's certainly worth reading. Now, even though I started out my talk by picking on sticklebacks in a way, I want to point out that even the individual who has done more than anyone else to essentially expose the world to this what we can learn about reproductive isolation from sticklebacks, Dolf Schluter is very much an advocate of the idea of a more complex and nuanced view of speciation. So even though Dolf Schluter would probably typically be associated by many biologists as thinking about reproductive or the ecology of speciation as largely being about the study of reproductive isolation and adaptation and how they relate, he's been developing this idea of what he calls metapopulation dynamic controls on speciation, which is much more similar to what Mayr was describing in his book. And so, for example, in this AMNAT paper from 2016, he describes that increases in the speciation rate can actually happen either because populations evolve reproductive isolation quickly, or because they have increased demographic rates governing what he calls the metapopulation dynamics of speciation, by which he means the frequency with which spatially separated populations become established, the probability of population persistence, and the speed at which species come back into secondary sympatry. So this is very similar to Ernst Mayr's general set of controls on the speciation process. Now, one of the key pieces of background for my talk today is that I'm going to assert and develop this idea that thinking about speciation rate and studying speciation rate is the key to understanding which of these controls are most important and for understanding their relative contributions to the overall speciation process in a general sense. So I'll give you an example here over the next few slides. So if we think about this hierarchy or this relationship of these processes that affect successful speciation, let's focus on reproductive isolation for a moment. So if we think about speciation rate being governed by reproductive isolation, what we're really predicting is that if some lineages can evolve reproductive isolation faster than other lineages, then all things being equal, if reproductive isolation is the main control on the rate at which speciation happens, then lineages that evolve reproductive isolation fast will also speciate fast. Lineages that evolve reproductive isolation slowly will speciate more slowly. So you can imagine graphing it. So you've got on x-axis the rate at which reproductive isolation evolves, y-axis the rate at which speciation occurs. And this is the way that we already conceptualize the action of things like sexual selection on speciation. So this is not an idea that originated with me. So this way of thinking about reproductive isolation and speciation rate is already well established in the literature, especially in areas like the relationship between sexual selection and speciation. And so if we go back to this, there's a classic paper by Mary Jane West Eberhard where she basically predicts this sort of relationship, and she talks about how basically sexual selection that is stronger in some lineages is basically going to lead to more significant pre-mating reproductive isolation between taxa, which is also going to incidentally then lead to fast rates of speciation. So you might imagine that you have species that have weak sexual selection, but they're going to have reproductive isolation evolve relatively slowly. And you might imagine that species with strong sexual selection are going to evolve reproductive isolation quickly. And all things being equal, that should translate into effects on the rate of speciation as well. So with that background aside, I'm going to tell you 3 stories today that bear on how we've been testing aspects of these controls on speciation itself. In the first part, I'll review some older work we did a few years ago looking at the relationship between reproductive isolation and speciation and talk about how we can test these general kinds of ideas. And then I'll talk about some more recent work we've been doing on population isolation and speciation rate in irradiation of Australian lizards. And then I'll talk about some work that is still preliminary that we're doing on a more general view of population isolation and speciation rates using neotropical lizards and snakes as a model system. Now, I have just a little bit more background to go through because I want you to I don't want to develop some aspects of the methods behind some of the things that we're doing in this talk. And so I just want to get you in a mindset of thinking about the speciation rate in the way that I'm going to be describing it during my talk. Because I'm not going to go into details about how we're estimating speciation rates. We're just going to take them as something that we estimated, and we're going to do things with them. So let's say we've got a time-calibrated phylogeny for some group of organisms. In this case, these are parulid warblers. And the sorts of things that you're going to see in this talk and the sorts of methods that I'm going to be using to get the rates that you're going to see are based on taking a tree like this and fitting some statistical models to it and turning it into a picture of speciation rates across that tree. And so this particular example is an estimate of speciation rates across this phylogeny from a program, BAM, which was developed by my lab. The key point here is just that the branch colors on this phylogeny are reflective of the model predicted rates of speciation on this tree. This is just one way of estimating speciation rates. And the specific speciation rates that I'm going to focus on today are going to be what I will call tip rates. And these are, think about this as the time-calibrated phylogeny. I'm going to focus on speciation rates at the tips of the tree. So when I say speciation rate, or when I say tip rate, what I'm really talking about is you might say, well, how can a species have a speciation rate? Well, you think about a tip rate for a given species as just an estimate based on past evolutionary history of our best estimate as to what the present day or instantaneous rate of splitting of a given lineage is. You could sort of think about it in a future sense of like, hey, if I take American robin and I know its speciation rate, that's just a model-based prediction based on the phylogeny of that family of birds that says, hey, on average, I sort of expect to wait how long into the future before that lineage would split if current conditions continued into the future. So we can plot speciation rates for each individual tip. Like you see here, I can pull out rates for individual taxa and show you them, like I'm showing for yellowthroats and redstarts and other species right here. So you're going to see a lot of this throughout the talk, but I'm not going to talk at all about the methods. Happy to talk later about that sort of stuff. And the other thing to know, because the other question that often comes up is how well do these methods, whether it's BAM or other methods like what's called the DR statistic or the node density metric, these are other methods for estimating speciation rates. So how good are these methods at estimating speciation rates? So it turns out that they actually are pretty good. And there's some theoretical reasons to think that we should do very well at estimating speciation rates close to the present. And so this is just a paper published by my former graduate student, Pascal Teitel, this year, looking at how these different methods, BAM and DR and node density, have performed at estimating speciation rates given some simulated data sets and so on. And the only thing I want you to take home here is that these methods do reasonably well at actually estimating speciation rates. And for the paleontologists in the audience, because this comes up all the time, I want to emphasize that these really are speciation rates, not net diversification rates, not worrying about extinction, actual rates of origination, speciation. So we can talk about that after as well, but these are rates of speciation and not net diversification. Now the final bit of background you're going to see is that I'm going to show you some analyses of the relationship between specific traits and speciation rates. And so you're going to see some plots that look like this. Now, the only thing you need to know is that when I show you those kinds of plots where I show you a trait and a rate of speciation, you can know that I'm using special statistical approaches to control for all sorts of things that are going on in the background, from how speciation rates shape phylogenetic trees to non-independence between taxa in the analysis. And so we've done a bit of work on methods for this, and along with a number of other researchers from other groups have developed other sorts of methods. This is a very active area of research. So let's go back to this set of general questions that we're going to look at. And the first thing I'm going to talk about is the relationship between reproductive and isolation and speciation, to try to ask, is reproductive isolation, or the rate at which reproductive isolation evolves, a key limiting step in the speciation process? So let's think back to this sort of control interaction network. We've got reproductive isolation, ecological differentiation, formation of isolated populations. We're going to focus on reproductive isolation. So to do this, we need to test the relationship between reproductive isolation and speciation and this framework that I'm describing. We need two things. First of all, we need to be able to estimate the rates at which individual lineages evolve reproductive isolation. And the second thing we need to do is to estimate rates of speciation using phylogenetic trees. Now, there's a reasonable amount of data for a reasonable number of taxa on reproductive isolation. So what does reproductive isolation data look like? So I'm only going to give you one example. This is one example that could have been plucked from dozens of such studies on taxa that range from Drosophila to African cichlids to North American darters to a number of plant groups. But what's typically done is some researchers will go out and take some organisms, and they will do a bunch of crosses, and they will look at the fitness between species and look at the fitnesses of the resulting hybrids. And so this is an example you're going to see involving birds. Most of this data is actually it's a sort of a based on a compilation of hybrids and their fitnesses going back to the 1950s. And what you're going to see on the next slide is the relationship between, every data point is going to be a cross between two species. And on the x-axis is the genetic divergence between those species, and on the y-axis is a measure of the fitness of the offspring of those hybrid crosses. Now, if the hybrids are perfectly fit, they basically have a value of y equals 0, so they're fully fertile, so down near the bottom. And if they have a intermediate fitness where maybe they're simply infertile, they will be around halfway up the actual y-axis. And if both sexes are inviable, so all the offspring die, the cross simply fails, then we have complete postzygotic isolation, and that basically would score a 5 in this framework. So here's the data that's summarized by Price and Bouvier in Evolution, 2002. The only point here is that, and this has been published by a number of other researchers in other groups of organisms, the only point is that reproductive isolation, in this case postzygotic isolation, increases with time since divergence. And what this basically means is that biologically, that when you take two populations that are spinning off independently on their own trajectories, they're basically accumulating mutations that when those things are brought back together into a common genetic background, they cause a reduction in fitness, potentially an ultimate reduction in fitness such that the offspring fitness is 0. And we know that this scales with time since divergence. This has been shown in a wide number of taxa. Now, the trick that we're going to use to estimate rates at which reproductive isolation accumulates is to try to test how different groups of animals Specifically, birds actually accumulate those essentially genetic isolating factors that contribute to postzygotic isolation. So what you might imagine is that some clades of animals might have what we think of as a slow accumulation of reproductive isolation with respect to genetic distance, and others might accumulate reproductive isolation very quickly. And so this is work that I'm going to talk about. This work I'm talking about now was done in collaboration with Daniel Matute, who's a professor at UNC Chapel Hill. He gave a talk here last fall. And so we developed a theoretical framework and a statistical model for estimating the rates of reproductive isolation from these types of pairwise cross data. And we applied it to birds, we applied it to Drosophila. And here's an example of an empirical example from birds showing the differences in the rates of postzygotic isolation accumulation for a couple of clades of birds. So up at the top, you have one clade of birds, the pheasants, which accumulate reproductive isolation relatively quickly, and they're in red. And you can see that because the slope, these data points are scaled on a common y and x axes here. So you can see the slope of that accumulation, that's the fitted slope of that accumulation of postzygotic isolation with respect to time, is steep. And on the bottom, we have another clade of birds, the parrots, which accumulate reproductive isolation relatively slowly. Now, just to put this in sort of commonsensical terms, the way to think about this is that on average, if you took two species of pheasants and you crossed them, they probably would not produce viable offspring. However, parrots with a slow accumulation of reproductive isolation, you might be able to take even very divergent species and cross them and still have viable offspring, potentially even fertile offspring. So we can take this model, we can estimate parameters which basically are nothing more than the slopes of these relationships with some tricks that we use to correct for non-independence and so on. And we can estimate this tempo of reproductive isolation evolution for all these different groups of birds. So this is just showing you this distribution of postzygotic rates, if you want to think about it like that. And so we have everything from things that evolve it quickly, they're poor at hybridizing, to things that can effectively hybridize with lots of other things. And so those will be good hybridizers. So that's how we estimate postzygotic rates of postzygotic rates. Now we also have speciation rates for birds. So what I'm showing you here is just a rank order plot of speciation rates across 10,000 species of birds. So all you need to know here is that point is just that we have estimated speciation rates using a comprehensive phylogeny for birds. Details don't really matter. The rates go from things like New Zealand riflemen, which have slow rates of speciation, up to things like ducks and geese, which have relatively fast rates of speciation. And the test that we're going to do here now is pair these things. So we've got rates of reproductive isolation evolution, rates of speciation. We pair them together. We're going to do a test. The prediction, of course, is that if postzygotic isolation in this sense is a key limiting control on speciation, they should be positively correlated. So what are we going to see? Noise, effectively. So we don't really get much of a relationship here between postzygotic rate and speciation rate. In fact, the slope of this relationship is actually negative. It's non-significant. It doesn't matter how we estimate speciation rates, we get essentially the same result. And so the key take-home message here is that the rates are decoupled. So we have clades that acquire reproductive isolation quickly. We find that they do not actually speciate more quickly. And we've also gone ahead and looked at this across a number of other clades, specifically Drosophila, number of groups of Drosophila. And we've also looked at prezygotic isolation in Drosophila using some classic data on this from Jerry Coyne and Alan Orr from some studies they did in the 80s. And in each case, we find no effect of the rate at which reproductive isolation evolves on the speciation rate. And there's a lot of limitations of these analyses, so I don't want to get into too much about these analyses. Reproductive isolation is inherently noisy data, especially for birds. It's very noisy. In the case of Drosophila, the phylogenies that we're using for speciation rate estimation are noisy. But the point is that even after applying these things and nonetheless thinking about these limitations, it's still true that we don't have any evidence that reproductive isolation is the rate limiting control on the speciation process. And so what I'm going to move to now is thinking about this framework that we use for addressing reproductive isolation as a control on speciation, and try to look at some other possible controls on speciation. So now we're going to move to if we can quantify the rate at which populations diverge, and look at how it potentially predicts speciation rate. And we're going to focus here on irradiation of Australian lizards. Now, this idea that the rate at which populations diverge, and if you can think about species range, laying on top of a continent. And you can think about the rate at which populations diverge as basically the rate at which some of those populations are accumulating genetic divergence relative to other populations, perhaps as a function of landscape heterogeneity or weak geographic barriers and so on. This idea that that type of divergence predicts speciation actually goes all the way back to Darwin. So if you read the Origin, chapter 2, you'll find that Darwin talked quite a bit about this, what has become known as the manufactory hypothesis. So it's not a super well-known hypothesis, but it's a little interesting part of chapter 2 where Darwin really gets into one of the most macroevolutionary themes in the entire Origin. And so what Darwin's idea is that he predicts that there's this continuum where species within species are always producing incipient species and varieties within species, and the species that are producing the most varieties are also the ones that should have the most species at a higher level. So he predicts that there's this scaling of speciation and divergence at all scales. So he writes in the Origin, he says, if the manufactory of species has been active, we ought generally to find the manufactory still in action. And as a general rule, wherever many species of a genus have been formed, the species of that genus present a number of varieties, that is, of incipient species. So basically, if you have something that has lots of speciation, it should also have lots of incipient species or lots of divergence at the subspecies level. And so the rate of species formation in this framework should correlate with intraspecific divergence measured somehow. So we're going to test this idea using one measurement of divergence between populations. And so we're going to think about ways, how can we estimate the formation of population isolates? So we're going to get estimates of population divergence for individual taxa, and then we're going to estimate rates of speciation using phylogenetic trees. We're going to bring those together just like we did in the examples using reproductive isolation in the previous section. And I would acknowledge here my former postdoc, Sonal Singhal, who's now a professor at California State University, Dominguez Hills, who's really taken the lead on a lot of the analyses in this section and in the next section. So the system that we're going to apply this to is Australian sphenomorphine skinks. So this is Australia's most diverse vertebrate radiation, about 250 species of lizards. They have a lot of variation in body form, from fully limbless things to things that look like very typical lizard forms, lots of pretty color patterns and so on. This is a group that I've been working on since about 2004 when I started my PhD. And one of the things that we've generated for this group is a time-calibrated phylogeny for the sphenomorphines. So it's complete at the species level. Doesn't really matter for the purposes of of my talk now, but we have speciation rate estimates across this phylogeny as well. And one of the interesting things about this group is that there's a pretty strong shift in speciation rates within this group of skinks. So we have one clade, which basically is defined by that arrow, which has undergone an increase in the rate of speciation. So they have faster speciation than the sort of outgroup lineages at the top of this figure that have a bunch of blue towards the present. Now, if population divergence is a key rate-limiting control on speciation, we can make some predictions here. One of the things that we can predict is that the parts of this tree that have slow speciation should also be associated with slow population divergence. In other words, their populations should be more connected on average. They should be differentiating less into incipient species, and thus, at a higher macroevolutionary level, we see slow rates of speciation. Now, there's also this big clade of fast speciating things. So we predict that this group should have relatively fast population divergence, fast speciation, and their populations are going to be diverging faster in space. They're going to be forming incipient species at a faster rate. Now, how do we do this in practice? Well, we get range-wide sampling for a bunch of species of lizards. Here's a distribution of samples for a single individual or a single species, Ctenotus robustus. And we use double digest RAD sequencing, got about 2.5 megabases sequenced per individual, about 59,000 variable sites per species. So we've done this for a bunch of samples, a bunch of species. And then we're going to calculate the pairwise genetic distances between individuals. And we're going to look at the relationship between that and the pairwise geographic distance between individuals. And we'll get a relationship that might look like this. What does look like this in the case of this particular species. Now, each of those data points is a pairwise distance between two individuals. And we can estimate the isolation by distance for that species using that data right there. Now, species that have a high slope are diverging faster in space, and species that have a low slope are less isolated by distance, and they're diverging less, and they're less divergent with respect to space. Now, overall, in terms of sampling, So we have a lot of samples. So there's sampled 892 individuals, about 75 species. And because I'm here, and because I have your attention, I can ask this question of where all these samples actually come from. So I think there's sometimes this perception that when we do these kinds of studies, show up in evolution, whatever, that getting samples is a little bit like ordering a pizza. So just call up Michigan Herpetology and ask for some samples, and the samples just come. And they're free samples, of course. Now, it turns out that getting samples like this is actually challenging, it's expensive, and it's logistically hard. So there's a paper that was published a couple years ago that actually estimated how much it costs on average to get a single sample from a tropical snake. That number is about $120 US. That's about what we pay to get samples from tropical snakes. Those skink samples that I referenced on a couple slides ago, many of which were collected by us, many of which were collected by partners at other museums, probably cost a total value of expeditions to collect those samples is probably millions of dollars. So, and this is just to put some of this in context. So this is since 2004. These are places that I have personally spent at least a week and in some cases several months doing active field collection. And it's quite difficult to collect reptiles and amphibians in the field, so you actually have to spend a lot of time in particular places to get the really hard to get species. And so this basically brings us to lots of really remote places where we end up doing a lot of field work in places like this, up in the northern Great Sandy Desert. Here we're up to several hundred miles from the nearest paved road, nearest power source, and so on. And the work is very hard. It often involves teams of people to do things like putting pitfall traps into very rocky soil, like you see here. And so through this all, we've had a lot of help from folks here at the University of Michigan. You recognize some of the faces in some of these pictures. And we've also had a lot of help from other folks. So these are us, and this is on a 2015 expedition with members of the Martu Aboriginal community in Western Australia. Now for the next section of the talk, where I'm going to talk about neotropical stuff, a lot of our samples came from Brazilian collaborators, and a lot of samples came from work we've done in Peru. And so for the last few years, many of you know that we've been working in the Peruvian Amazon. So this involves going to hot, wet, buggy, and remote places. And so we've had a number of really great field teams involving a lot of folks from the University of Michigan. We got these are our two of our 2016 field crews, and these are our field crews from 2017 and 2018. And of course, we also have a lot of contributions to projects like this and others from other folks affiliated with Michigan Herpetology. For example, we now have a number of University of Michigan students who've been going off and leading and participating in expeditions, even independently of the actual curators in herpetology. And then we, of course, have colleagues in Brazil and Peru and Australia who are also actively collecting. And of course, I also have to mention that with all these specimens like this that we get, there's another very important aspect of these specimens, which is yet another cost associated with these specimens, which is the curation side of it. And so, of course, we have an excellent collection manager here at the University of Michigan, Greg Schneider, who actually does a lot of the hard work of actually making sure all this stuff is properly curated and archived. And so this is just sort of my way of acknowledging the vast number of people who've participated in this, and the true number of people is in the hundreds. Now, these are our University of Michigan participants that we've had over the past couple of years, and many, many organizations, which you see on the right, who've contributed individuals and logistics to some of our work. Now I'm shifting back to the science side of things. So here we are, isolation by distance for a particular species. Now, one of the things that we see when we start to look at these plots is there's a fair amount of cryptic diversity that pops up in some species. And this is a typical kind of pattern that you get with these kinds of data, where you plot geographic distance against genetic distance and you get two clusters like this. Now what these clusters correspond to in this case is basically a case where this species had essentially two genetically distinct species within it. So you have within species distances and a between species distance. So we use a number of different ways of getting at that problem by doing some different forms of species delimitation in our analyses, including using traditional taxonomy along with using coalescent-based species delimitation. Now, if genetic estimates of divergence that we're going to discuss here are correlated with organismal traits associated with population isolation, we might expect to see if our divergence estimates that we're getting for species are good or useful, we might expect them to correlate with traits that we think are correlated with divergence, morphological traits. One of those traits that's been speculated about is body elongation or limb reduction, where the prediction is that species that species that actually have reduced limbs, lizards that have reduced limbs, are going to be more limited by their substrate because they're burrowing animals, and they're going to diverge more in space. So we've tested this relationship by looking at how within this radiation, because we have species that look like normal lizards, species that look like largely limbless lizards, and we've looked at how genetic divergence scales with the limb reduction of those individual species. And when we do that, we get a relationship like this. This is a significant relationship. It just tells us that species that are limbless burrowers are more divergent in space, population genetically speaking, than species that look like typical surface active lizards. So that's sort of a nice check on whether our genetic estimates are correlating with things that we already sort of think are correlated with the ability of organisms to disperse across a landscape. So, does population divergence predict speciation rate? No. We basically get, if anything, we have a weak negative relationship here. And so we really have kind of a mess of points here. So we have no substantial relationship between speciation rate and population divergence. This is very similar in some ways to what we saw with reproductive isolation. Now, another way of looking at this, so if I take those population divergences and bin them by the major clades within our data set, there's basically 3 major clades within this data set. And you can see this group called Tenotus, which is the light blue group of points, that has those species tend to have relatively low population divergence. Now you'd predict that given that they have low population divergence, they would also have slow rates of speciation, because their populations are more connected and they're forming incipient species at lower rates. But in fact, what we see is that Tenotus is actually a group that has both fast speciation and low divergence. So this is a concrete sort of visual showing us that this runs opposite our overall predictions. Now, conversely, we have this quasi-clade here, where we have slow speciation and high divergence. So again, this is sort of opposite what our overall expectation. And so just to summarize this section of the talk, so we estimated population divergence rates across 75 species from Australia's most diverse vertebrate clade. We found that limb reduction, which is kind of a dispersal proxy, predicts population genetic differentiation. But we didn't find any relationship between intraspecific differentiation and speciation rate. So this brings me to my final section of the talk, which is the relationship between population divergence and speciation rates in neotropical lizards and snakes. Why am I focusing on neotropical lizards and snakes? Well, one question you might have asked, given the last setup with those skinks, is do we have enough phylogenetically independent variation in speciation rate to even see an effect within this clade? So this is the skink phylogeny with speciation rates mapped, and most of the speciation rate variation is just partitioned between 2 parts of the tree. Basically some outgroups, a rate shift, and then some big clade that has basically one relatively fast rate. So there's a lot of reasons why this might not be a good representative system. There's just not a whole lot of, this is effectively a system with just a couple of phylogenetically independent data points. And so to get around this issue, or try to expand our analyses, we've been looking at divergence across lots of clades of neotropical squamates that actually differ in their rates of speciation. And so we're characterizing range-wide population genetic structure and ecological divergence across about 100 species from Brazil and Peru in particular, so from the greater Amazon and from the Cerrado and a little bit of the Caatinga habitats. So these are some representatives of some of the taxa that we're looking at across that region. And so you can see here, these are generally fairly unrelated taxa. And we have estimates of speciation rate across squamates in general. And we can basically take this analysis and map these speciation rates onto pruned phylogenies with just 100 of our focal taxa. And we've been estimating population genetic divergence from a set of markers that was developed in my lab by my former postdoc, Sonal Sengel. And so the details here aren't important, but basically we've been generating very rich population genomic data for each individual. So we get about 4,700 loci per individual, about 3 megabases of sequence at 10x coverage per individual. And so we have about 100 species, 1,800 samples to work with so far, and the results I'm showing you now are just based on the first 46 species that we finished. So this is preliminary stuff here. So this again is just showing you these relationships between genetic divergence and geographic distance and genetic divergence for a couple of random species of snakes from this data set. So you can see these nice isolation by distance relationships here, relatively low slopes. Now, low is relative in this sense, and the reason I'm showing you these is because one of the interesting things that we found in doing these analyses is that there's a really nice signal of difference between that separates the effects of geographic distance on genetic divergence for lizards and snakes. So the left column here are 2 snake taxa, and the right column here are 2 lizard taxa. Now you can see that the steepness of those slopes for the lizards is much steeper, indicating they are proportionally much more isolated with respect to distance than the snakes. That's kind of interesting, and it was a bit surprising in some ways because lizards in general, lizard taxa typically have far higher population densities than many of these snakes, so it wasn't necessarily clear that this is what we were going to find. And so just looking at what we have overall in terms of the relationship between speciation rate and population divergence, so once again, we basically find no relationship between speciation rate and population divergence. If anything, this is a trend in the opposite direction to what we expect. Remember, we're expecting to get a positive relationship between the rate at which populations are diverging and the rate at which speciation is happening. We do not see that. And just kind of breaking this down by family here, I'm color coding the points by the major taxonomic group to which those things belong. You can see again that what I was showing you about snakes having relatively low differentiation, so they're clustering down here near the low end of the x-axis. And in terms of speciation rate, snakes have fast speciation. So snakes are actually bucking the clade is really bucking the trend here with a prediction. So we have low differentiation and fast speciation within snakes. And so just to summarize this section of the talk, so we estimated population divergence rate as we're thinking about this isolation by distance across range, across about 46 species of neotropical lizards and snakes. And we have lots of in contrast to the Australian sphenomorphine skink clade, here we have lots of variation in the speciation rate. And our clades vary a lot in population genetic divergence as well. So for example, snakes tend to have relatively low divergence. But despite all of this, we find no relationship between intraspecific differentiation and speciation rate. So this is concluding the 3 main sections of the talk. And just to wrap up, I want to bring you back to that original question that I asked, which is what is speciation? And how do we think about, how do we think about speciation, and how should we be thinking about speciation? So we think about speciation often like this in traditional evolutionary biology, where we're focusing a lot on microevolutionary studies that are telling us about the evolution of reproductive isolation within relatively recently diverged populations. Now, this is where most speciation research is concentrated, and I would say that we're getting amazing insights into reproductive isolation and population divergence from these kinds of studies. And let's not underestimate how important reproductive isolation has to be on some level. If you didn't have it, species would not exist because they would collapse back together, right? So even if reproductive isolation is not the rate-limiting key control on speciation, it has to nonetheless be important. You don't have species if you don't have it. So that's important. But I would say that these kinds of studies are providing us with very little information about the importance of those processes for successful speciation, the kind of speciation that goes on to contribute to the birds that are breeding in the woodlot behind your house. And we have very little understanding as a result of this work about how we end up getting this population of speciation events. So that's the sort of what we're getting out of microevolutionary studies. So what are we getting out of macroevolutionary studies? Well, I think phylogenies, and I will argue that the one thing that phylogenies are really good for is giving us a great pattern-based perspective on what speciation rates actually are in nature. This is something that would have probably shocked Ernst Mayr in 1963. In Animal Species and Evolution, Mayr writes, it's probably the case that we will never understand speciation rates and how they vary in nature. And I would say that nothing could be further from the truth now. So if I was to think about things we really have learned in the past 10 years of evolutionary biology, it's how much speciation rates actually do vary. That said, I would say that phylogenies are only weakly informative about underlying processes. And there's a lot of interest right now in the field in developing sophisticated models that you can fit to phylogenetic data that are supposed to tell us about these underlying processes of population splitting and persistence and reproductive isolation. And I don't think we're going to get any major insights into those lower mechanisms from simply fitting better models to phylogenetic trees. I'm very skeptical of this. And I think we are very quickly approaching limits to knowledge in terms of what we can gain from these types of phylogenetic diversification models. So it was a nice paper that was published last year by Jing-Chun Lee, who did her PhD here with Dearmet a few years ago. She's now a professor at the University of Colorado, along with some folks from Lacey's lab. And they were looking at some of the ways in which basically what kind of macroevolutionary patterns do we get from some of these different microevolutionary processes. And one of the satisfying conclusions from my perspective from this paper was that they found, so they were looking at what you're seeing here on these graphs are different parameters for population processes, like population splitting rates, rates of evolution of reproductive isolation, and so on. And what they found was that very different combinations of parameters, fundamentally different parameters, could give you the exact or similar phylogenetic diversification patterns. So that suggests that when we were trying to extract information from phylogenies alone, we're going to hit a wall. And fundamentally, some of these processes are going to be non-identifiable at that level. So how do we link microevolutionary patterns to microevolutionary processes to phylogenetic patterns? Well, I think that the framework that I sketched out earlier, which didn't find any significant results in any clades, nonetheless has some promise. However, I think there's still a lot of caveats here. One is in the case of the analysis between divergence and speciation rate, is population genetic divergence in terms of isolation by distance the right thing to be looking at? Maybe it's not a good measure of the rate at which incipient species form. So there's dozens of caveats one could list here for the conclusions that I've drawn in those latter two parts, in the second two parts of my talk. In terms of the reproductive isolation stuff, we could ask, did we study the right type of reproductive isolation? Maybe if we studied a different type, we would get a different result. And how robust are those reproductive isolation data? Do we even have the power to really robustly estimate those parameters? So there's a lot of things to be said on that. I don't have time to go there. But the one thought I want to leave you with is sort of my prediction about what I think maybe the most important process is, and it's the idea of persistence. And I think that persistence of populations has to be one of the keys to explaining biological diversity in a general sense. And the sticklebacks kind of make this crystal clear, because we clearly have a species of organism that is capable of rapidly diverging into numerous differentiated, ecologically differentiated populations that have some measure of reproductive isolation. Yet over geological timescales, almost all those populations are just microevolutionary ephemera. They essentially go nowhere. Somehow you get hundreds of populations in 12,000 years, and you only get 20 or so such populations that have the persistence to last over 20 million years. So that's a very remarkable sort of thing. So what can we actually do to study persistence? This is a very hard thing to study. So if I was to propose what sort of solutions I see on the horizon to cracking this problem, it's going to emerge from a novel fusion between phylogenetic speciation patterns, which I think we're quite good at doing now, and a bunch of non-traditional data types that we can apply and use in the sort of framework that I described here. And I think there's a lot of things that we can bring to bear on this problem, from population ecology and conservation biology, quaternary paleoecology and global change biology, and other sorts of things that involve novel traits about demography that we can stick on the tips of a tree and ask questions about what's driving speciation rate variation. So just some specific examples here would be applications of population viability models in a comparative phylogenetic framework. We have experimental demographic data from lots of species that can be leveraged to test these sorts of hypotheses. There's subfossil and paleontological studies on prehistoric range shifts and species extirpations from particular regions. And then we have biodiversity data from museum specimens, where we actually know things about how historical range shifts from actual species occurrence data relative to where species occur now. And we could leverage that in a phylogenetic comparative framework to maybe test some of these hypotheses. And so that's where I'm going to end my talk. So there's lots of acknowledgements here. I want to thank my collaborators on this spatial work in particular, especially Sonal Singhal and Gabriel Acosta for their contributions to the data analysis and sample procurement and everything else, and especially in terms of funding, the Packard Foundation and NSF and University of Michigan. And thanks for your attention. If I have any time, I'm happy to take questions.
Thank you. We've got time for questions, and we have this microphone. Okay. Who's first? George.
So when you measure speciation rate, I suppose it's per million years, the unit, right? Number of events per million years. But when you measure the
when I'm measuring speciation rate, what?
What's the unit?
What is the unit?
Yeah.
The unit is the per capita rate of lineage production per million years.
Yeah.
It's an instantaneous rate per time.
Right. When you measure the rate of reproductive isolation, the formation of reproductive isolation, I noticed your unit was per genetic distance, unit of genetic distance. So if different lineages have different mutation rate, then the genetic distance does not translate to million years in the same way. So how does that affect your result?
So that's possible, although when we did the actual analyses here, you would have to have, so when we did the actual analyses here, I'm using genetic distance as a proxy for time, or because in the case of the bird data, that's the original figure was in units of genetic distance. The analysis of birds was actually done in terms of a time-calibrated phylogenetic tree. So to the extent that you believe that you can correct for those differences in mutation rates to get things onto a common time scale, that would take care of that, because we are analyzing it not in, all the analyses that I did there assumed that we could actually linearize those trees and make them ultrametric. So we're trying to correct for lineage-specific differences in substitution rates. So not to say it's perfect, that's certainly one source of noise that could potentially bias both rates of speciation and your estimates of pairwise divergences to screw up your reproductive isolation rates, right? So that's just sort of that's like another layer of assumptions that somebody else has to fix. So.
Well, because you got negative results, that's why noise matters.
No, this is true. I mean, we've done a lot of power analyses here to basically say, okay, so one of the things you could ask is, how strong would a relationship have to be given the amount of noise? So we've estimated the amount of error we have in all those things, and we've done some power analyses with those. Yes, we have, I would say, medium power. We certainly don't have strong power with these kinds of things. There's no getting around that. Some of the reproductive isolation data, especially for birds, really suck. Actually, the speciation data I'm much more confident in some ways than the reproductive isolation in terms of its error contribution to the total. We could kind of flip the power question around, though, a little bit and ask, for example, given that we actually have negative slopes out of some of those relationships, you could also say, rather than, how strong does the effect have to be to get a significant effect, you could also say, how likely is it that that you actually had a true positive effect while nonetheless observing a negative effect? When we do it like that, that actually ends up being, it suggests we actually would have had quite a bit of power in terms of not seeing the sort of negative relationship we observe overall. But I agree that power is absolutely an issue, so we can't rule that out. We can do some things we don't even know in some ways how much error we have in the reproductive isolation estimates. I mean, you've seen how messy some of these kinds of data actually are. But that's what we have to work with, and it's a lot of work to actually get those data. It's like 80 years of pairwise crosses in labs for Drosophila,
Yes, you do. Here.
So do you think that speciation rate has an influence on how likely you are to observe a lot of genetic differentiation? So if something has a really fast speciation rate, maybe you don't see a lot of genetic differentiation in those species because they speciate so quickly? Does that make sense?
So I agree that that's possible for some kinds of taxa that are super rapidly speciating, but the thing is, the speciation rates we're talking about here are all basically so slow that the genetic structure is all happening on a time scale that is far more recent than that. So I don't think that's actually a confounding factor. There are different ways we might measure differentiation that would not be robust to that assumption, though. But that specific measure that I'm talking about here, I don't think is affected by that. That said, I do think that there's one of the sort of caveats is that, so when I talk about differentiation, one of the things I do think is bound up in differentiation is I don't think it's actually estimated independently of persistence. So I think that the rate of lineage turnover on population turnover on the landscape does feed back into the sorts of divergence estimates you can get. So they're not fully independent. So there is that.
So I very much agree with you that we should start looking at the species persistence. But are you actually doing something on that, or if you can develop more about? You mentioned all these hypotheses and data that can be used. I would add to that modeling, network modeling. But okay, go ahead.
So are we doing things with the persistence side?
Yes, that's the question. Oh, and what you're doing.
In terms of my last slide there.
Exactly.
Yes, but we are this far down that actual road yet, but
okay.
So.
And what kind of data you using?
I have, so I'm thinking about how to answer this because I actually was on the fence between ending this talk on a positive note or a negative note. And so the positive note says, hey, we can use things like present-day range shifts, for example, to estimate how sensitive particular species are, and then say, hey, maybe that's a proxy for how well they could persist over geological timescales. This negative side, or the skeptical side, would say, like, oh, come on, there's still another layer of assumptions here. You're going from anthropogenic disturbance, essentially, even though, over the past couple hundred years, those shifts, then you're going to assume that tracks the sorts of, that's the same thing that is a meaningful sort of geological timescale persistence. And I think you could make a good argument that it's not. You could also make an argument that that's the only kind of data we have to work with in some ways, so we should look at it. But you could also say that it is possible that there will be some fundamental limits in terms of what we can know about that area. I mean, so I think that actually I actually think there's a real limit to inference here, and I'm not sure where we're going to ultimately end up. So I think about what we get from a phylogenetic perspective on speciation, or the fossil record for that matter, tends to be on the time scales of resolution of speciation of events that are happening on the 100,000 year to million year time scales. And when you think about processes below that, either from phylogenies or from the fossil record, you have all kinds of things that relate to the quality of the fossil record, time averaging effects, the noise in divergence time estimation from phylogenies, coalescence variance, all of which makes it really hard to do anything at a time scale resolution-wise that is finer than that. The flip side is that we also have we have a really good window into things that are happening on contemporary ecological time scales. In terms of we can study the ecology of populations in the present day, we can study patterns of genetic variation, but that doesn't actually tell us anything about how populations are persisting over that, let's say, 500 to 50,000 year time scale. So I think, and that's actually the time scale where I think the most important sort of metapopulation processes that relate to persistence are likely to play out. So whether some of those things at the contemporary time scale, let's say population viability models or experimental studies of demography and looking at species persistences on islands, you could do those experiments. Are those going to be good proxies for that 500 year to 50,000 year persistence? I don't know. I think it's worth doing it because I am completely stumped in terms of thinking about other things that we can do. So for lack of other things to look at, that's where I think we should concentrate efforts. So, but that's sort of the best I can do for now. But.
Well, you do have some information on how fast and far things have moved, because Michigan was kind of wiped out by a glacier not that long ago, and you know that some minimum distances things had to have moved, and approximately how long it took within the time scale that you're looking at. So
I definitely agree. So I think with some taxa, with subfossil data and paleontological data, in particular, like pollen data in particular, I do think that for a small number of taxa, that kind of analysis is actually going to be possible. And for some things, we have subfossil deposits on islands that actually tell us that species of birds or dwarf mammoths and other things were actually present, so we know some things about persistence. Though there's other things that one could criticize some of those things for, though. How much is a rebound from the last glacial maxima actually representative, though? Again, I don't know, but I agree with you that that's something to look at as well. So.
I have 2 points. 2 questions. So the first thing is that actually I'm kind of with you on your sort of intuitive assumption that if you have a lot of speciation, very high speciation rate, there should be high population divergence rate. But on the other hand, after I listened through your whole talk, and I see you did not mention anything about a mating system of these animals. Of course, I don't know enough about animals, because for plants, we have surfing species, outcrossing species, and their population divergence rate will be very different.
Well, this is true. So this is in some ways a complication. This is maybe something where if you were thinking about where a mating system actually plays a role in terms of that, so think about that hierarchy of controls that I sketched out. I think it would be a simplification to just say a mating system has one kind of effect within that paradigm. So I guess probably the accepted dogma, the typical way people would think about this in a macroevolutionary sense would be, let's say mating system is likely to affect reproductive isolation, because that's something that different mating systems we expect to drive different rates of evolution of things like species recognition characters, like gamete recognition loci, sexual signals, all these things that translate into pre-mating isolation or potentially post-mating isolation in some cases. Nonetheless, there's also some literature that would link things like sexual selection to actual demographic traits. So there's been arguments made that intense sexual selection can actually drive populations downwards towards local extinction. So anyway, I'm not really answering your question other than to say that arrow of causality between something like mating system and how it would affect different things from divergence to those macroevolutionary controls could be more complicated than we might typically think.
What I would wonder is that in your future analysis, if you tease out all these other factors to see if that pattern you expect from your assumption would come out in the end.
So I think that's a good point. I would say that in some ways, the scale at which we're conducting it for analyses actually controls for a lot of that. So like within the Australian skinks, there's effectively no variation in mating system. Maybe there's a little bit of variation in clutch size, but it's not the sort of grand scale of life history variation that we know explains a lot of patterns of like divergence between taxa. So if you look at like patterns of genetic variation across like the tree of life, for example, most of that divergence and that variation, it ends up being explained by really big differences, like massively R versus K selected species, for example. So continuum of like things that produce like hundreds of thousands of propagules to things that invest heavily in a small number of propagules. That has a big effect on genetic variation. But at the scale we're looking at here, we're like that life history variation is constrained to like this much, because we're talking about like squamates. The further we expanded scale into where I'm starting to look at generalities across a combined analysis that spans plants and vertebrates and arthropods, then I would definitely have to think about that more. I think I would have to worry about it a lot more and think about it as a covariate that could drive some of the variation.
Anyway, so the second part I have questions on the last part of your talk. So there, for that big phylogeny, I think people have been doing it for the last 10 years and try to look at it in a way sort of simplistically, look at phylogeny to see what pattern will come out. But now what I wonder is that if you plot these environmental variables, how fast ecological factors change, that may affect your speciation rate.
One of the things I would say is that the speciation rate estimates are noisy, but they're relatively robust. When I take squamates and I look at the estimates of speciation rate that come out of, say, the last 5 incarnations of a phylogeny for squamates at that scale, they're pretty consistent. We've done a lot of things to look at robustness of that. One of the things I didn't tell you is that we're right now finishing a 10,000 species squamate phylogeny that's anchored with a really high resolution genomic analysis. So we'll have about 1,200 taxa for which we have 5,000 loci each. So that'll be definitely the highest resolution phylogeny in terms of an anchor for getting a better handle on those things. But personally, I think that it's useful to do, but I don't actually think it will change anything. So lots of data, no real change in rates. The basic picture will stay the same.
So just one question, then we'll finish it. Just a general comment, a great talk, by the way, in terms of the reliance on persistence. Persistence is clearly a very germane attribute in life. But if you are dependent on it to calculate speciation rate, clearly there are many lineages that do not persist. And I'm wondering, in terms of generalities, are there any comparable, to some degree, paleontological data sets where you get a more fully fledged picture of diversification, and what would that look like compared to the neontological?
So in other words, is there a paleontological data set where you do get a more comprehensive picture where it's fleshed out on that level? Yes. I would say whether it's general, I would argue that it's not general. So the best case scenario here is the one, so the best case scenario for the fossil record is one where you can imagine that there's continuous reign of things that get preserved in the fossil record, giving you a resolution at like 10,000 years or less. The one place where that happens around the world, preserving species across their range, is the marine microfossil record. So there has been some good work. People have not really taken this to the level of trying to say, well, I don't know if it might not work because we're talking about big pelagic organisms with large ranges, but there has been nonetheless very good high-resolution studies of speciation and environmental change, and then comparing those to molecular phylogenies using the marine microfossil record, especially foraminiferans. But you could do that with diatoms or calcareous nanoplankton as well. So there's the potential for that sort of stuff, but then again, you have to think, okay, like unicellular photosynthetic things that have like massive population sizes that are basically pan-pelagic in that sense, and with very different things that you might not be able to say that much in terms of generality to land plants and so on. But the record is there.
Okay, well, thank you very much. Let's all thank Dan for